another video cast for the Radiation Research 2014 meeting. And before we start the video cast, I wanted to introduce a new member of the podcast team, Alison Burrell. And I will let Alison introduce our speaker today because it turns out they both worked in the same lab at the NCI with William Bonner, the person who discovered gamma H2X. So without further ado, Alison.
Hi, welcome. I'd like to introduce Asako Nakamura, who has also spent a lot of time in Dr. Bonner's lab and now has her own lab in, where are you?
Ibaraki University.
Ibaraki University. Yeah. And she's going to talk today about the poster that she has at the Radiation Research Society conference here in Las Vegas. Why don't you tell us a little bit about the focus
On
your
research?
poster?
Asako has done a poster dealing with Fukushima event, and they did something very clever. There were animals that were left wild in the Fukushima area, and they did some biodosimetry on those animals. So, Asako, why don't you tell us more about what you've done exactly?
Oh, actually, yeah. The background of this research is start like May 2011. Actually, government ordered to kill all the leftover animal. And we were thinking, just only kill the animal. Okay, we have to get all the information from these animals. And one project leader, the name is Dr. Fukumoto from Tohoku University, he actually asked me, why don't you check the DNA damage level by gamma 8X assay? And yes, it's right. And we did this project, we did this research, and we surprised we could see the gamma 8X foci, which means the DNA damage, and the cow's blood. It's already the first animal actually captured half year past accident. Half year already passed, but still we could see the DNA damage, which means this DNA damage assay is very sensitive, and we can get some more very important information in this project or research. That's a kind of overview of my research.
So, did you need to sacrifice the animals to get the blood, or did you just pull the blood out of those animals?
We killed them.
You killed them?
We did it, yeah.
And
because it's an order from the government. We have to kill them. That's the order, yeah.
But these were just farm animals?
Farm, yes.
Okay.
Yeah.
And did you use control animals that were far away from the Fukushima?
Yeah. Actually, we got the cow's blood. This one is not killed,
okay.
But cow's blood from Hokkaido. It's I think 600 kilometers far away from the accident. Also Tokyo. Tokyo is I think 200 kilometers far away from the plant. So we use these animals, these cows, blood as control. Yeah.
So tell us, how many damage do you see in the control versus the animals that were in the Fukushima area?
That's a very good question and a very difficult question to answer because my data is actually, our data is just a snapshot. Okay, we check the DNA damage level from the cow when we killed this at the time, We don't know the actual DNA damage radiation level at the accident. Okay, but I can tell you gamma 8X level, which we could detect it from the cow, is almost similar level at the 20 milligray acute radiation exposure.
So you're estimating the dose to be 20 milligray?
20 milligray, yeah.
But the exposure was mainly endogenous, right? It was happening through the absorption of cesium, or what was the compounds involved there?
The most of them is cesium. Yeah.
Cesium?
Yeah, cesium, yes.
So then you have one of the issues you're dealing with is once you ingest the cesium that is radioactive, how long does the cesium stay in the organism? Do you think it stays for a long time? Did you measure this in your work?
Not yet. But the half-life is almost 30 years or over 30 years of cesium. But other project team actually measure the cesium concentration entire body, like a bone and muscle or blood and blah, blah, blah. And the important point is that depending on the tissue, the concentration, accumulation rate are different. Okay, for example, cesium, they accumulate muscle. Very, 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 very highly accumulate coagulation in the muscle, but not in blood. So we have to think about the half-life depend on the tissue, or it's very difficult. We need a very, very difficult calculation, or also we need more information, not just only one side.
Well, you're actually lucky because cesium is a gamma emitter, and the gamma is a very long range emitter.
Right.
So
right.
you can make the assumption that the dose is pretty uniform, or at least you will definitely pick up the dose in the blood. So that's the good news. It's not like other compounds that would be a high LT compound. So that's in fact something you can use to your. I think it won't be as difficult.
So if the cesium is in the muscles of these cows, then you're seeing the results in the blood. So you were just testing the blood. You didn't look at the tissue at all.
No, not yet. But we still have a tissue in frozen tissue, or we keep all the tissue. So maybe in the future we can make some tissue section and we can check the gamma index acid in all the tissue. But it's going to be long and huge project. But we need maybe we can get the very useful information from this project, I think.
So you said something else when we were discussing earlier that was surprising. You said that you didn't see any spatial correlation. The proximity to the Fukushima didn't change the level of damage.
No, actually, I don't have a
Map?
but our I think we checked 70 cows in my project, but all 70 cows captured from the south side from the plant. And you can look at the maybe map and a Google search or something like a cesium contamination goes to the north side, west side. Yeah, the south side contamination is quite similar. It's not dependent on the distance from the plant. So that's why I couldn't see any difference between each area. Maybe these areas, the radiation dose are quite similar, not so big difference. It goes to the north.
Okay. Do you have access to other animals that have not been sacrificed yet?
Actually, next step is now we are sacrificing a monkey in this area. So monkey is very close to the human.
That's right.
So maybe we can get some more good information to close to the human response, maybe. But it's still ongoing project.
But
so.
these are wild monkeys.
Wild.
But I mean, I can understand why they're sacrificing the cows because maybe these are a source of food for the people. But why monkeys?
Oh, it's turned to be wild monkey, and they destroy the house in this area. You mean house, human house, because everybody are evacuated, and all the house and all the food for human food are left over in that area.
So the monkeys are pests.
Exactly. It's kind of
Yeah, but I mean, I understand the government's position, but how is it feasible to go through whatever region, distance from the center and really eradicate every animal that's been exposed?
I don't know that part. It's kind of political part.
Yeah, but it's interesting, the idea of pest. I like it. You can also go after rats,
Right.
because
Where are you going to stop?
And also the nutrition of those animals changes. So for instance, it'd be interesting to see if you see high level in the cows that have been eating the grass, which is contaminated, versus animal that would eat something that is not necessarily contaminated. And that will help the population know what is at risk for them and what is not so risky for them to eat, for instance. Is that the kind of strategy you're thinking of right now? And who's in charge of, I'm sorry we're changing you a lot, who's in charge of doing all these studies? Is there an organization that's actually in charge, or is it more like research study, grassroots movement type of thing?
That's a kind of organization.
Which organization is that? Which organization did you do this work under, for instance?
You mean government?
The government paid for this study?
Yeah.
Yeah?
Actually, yes.
So.
Yeah. And also, not only our project, there are so many projects. Of course, some projects actually working for the mice, checking the mice. And some projects are working for the fish, fish out very close to the plant. And also some guy are working the plant also, plant contamination or something.
Are they all using MH2X as a bio?
Not all of them, I don't think so. Like mice and I'm not sure, but yeah.
So we heard during this conference that gamma HX is a bad marker because it's not very. You lose the marker very quickly after the exposure. Obviously, what you're saying is actually it's not really true. That actually it is a long-lived markers in this kind of population.
But that point is my opinion is okay. I could see the foci, gammatic foci in a cow's blood after half a year the accident, but it doesn't.
After how many years?
Half a year.
Half a year.
Half a year. Even some cow 1 year after the accident, but still we could see the gamma-8X foci. But it does not mean these gamma-8X foci are made or came from the first impact.
Correct.
You know?
Right. So what could it be? What is the alternative hypothesis?
I think that came from one thing is a chronic exposure, right? Still there are high radiation. So maybe cow is outside exposed the very low dose chronic radiation, and another thing is that they are eating contaminated food, and also it's a both external exposure and internal exposure. So our future project or future point is we have to know what's the origin of these gamma-8X foci.
Right.
It's a very good, important point we have to answer.
Right.
Yeah. So that's the next step.
So if I had an advice, I would do two things. I would probably go into the zone and see if you can have different zones with different level of contamination and map it that way. But the other thing is really focus on different animals that have different nutrition.
Yeah.
Where you could tease out, the internal versus the external impact. I think it would be very powerful.
Yes.
And you said you still have tissue from the sacrificed animals that you could test.
It's very exciting.
Yeah. So in this case, we have to communicate. In this project, we have to share all the information, also all the we have to make some communication and can stick together all the data, and we can say something from this accident. We have to learn from this accident. Yes.
One last question on my side, at least. Very nice work. I think it's great. You're helping the community try to recover from this. And I want to finish on this topic actually. What's your feeling about the population over there? Do you think, and how do you dispatch this kind of information? Is it something you keep right now under research information and you don't dispatch it to the community, or is it something?
You mean community as
the people living in Fukushima or close to Fukushima,
person.
People who wants to return to this area? What is the rules or the protocol that you have in place for this?
Rule? We don't have a rule.
No?
Actually,
no rule.
No. If I got to this data, this work is ongoing, the preparation or publication. But after the publication, it's open, We don't need to regulate this information or something. It should be open for all the person as general person. But important point is general person does not have enough knowledge. Okay, let's say 20 milligram. What the hell is 20 milligram?
That's right.
We have to tell the right information, right knowledge to not scientific person, and what's the actual meaning of this dose or something. So there's no rule.
It's a good point.
Right, because it's important to explain to people who are not scientists this information in a much more understandable way. Because, for example, if this got out in the wrong way, that you're finding radiation in cows, then people might stop eating meat, and then it would have an economic effect on the the beef industry, I guess. So I definitely think it's important to explain it very well. Well, I think the gamma H2X assay is still coming into itself as a biodosimeter, and I think the more papers that are published, clinical papers where they're using human samples and showing a correlation to something, it will become stronger as a tool.
Right,
and,
right.
and then the government might be able to put something in place like that.
But you see,
But
what's
not now. Not now.
but any dosimeter, including a biodosimeter, needs a calibration. So for instance,
Right.
in our data, our control is at 0.2 foci per cell, and then the animal exposed at 0.5.
Right.
But we know, for instance, in human that you go from 0.25 to 0.5 within the time life. So if you don't control for age, for instance, you could, let's say, I'm going to play devil's advocate. The animals
Yes.
you looked at were older than you
Old
control.
cows.
And you're not looking at any
Right.
radiation effect, you're looking at an age effect.
Uh-huh.
So knowing the age dependency is critical. That's one point. But you can
Yes.
make this argument for even more things. You can make the argument that different people have different level, and therefore knowing where they were before is essential to be able to interpret the numbers. Because we're talking very small increments here.
Right.
So I think I'm a very big fan of the gamma H2X, but
But it's true, it's a very sensitive test. So it can detect a really small
Small change,
increase,
yeah.
change in radiation, so that you're right.
so
knowing
we all
the
agree as a
environment.
conclusion that the gamma H2X is here to stay and to help us protect the population.
Yes.
No, I'm very interested to see further work that comes from your lab on gamma H2AX.
Well, thank you very much for your time.